COVID-19 pandemic has challenged the most evolved species on Earth, that is we, the humans, and has shown us how vulnerable we are as a species. It has also been a stark reminder for us to realize the importance of science and the need of global cooperation to tackle global challenges. Namaste and welcome to yet another inspirational episode of Beyond Shodh. And today we are honored and delighted to have an exceptional scientist in the realm of molecular virology and immunology, Professor Arvind Sahu. Professor Sahu is currently the Executive Director in the Regional Center for Biotechnology, an institution at the forefront of biotechnological innovation in India. He also served the National Center for Cell Science, Pune, as senior scientist. Dr. Sahu's research primarily focused on the intricate relationship between the viruses and the immune system, particularly the complement system. Throughout his career, Dr. Sahu has received numerous accolades for his contribution to biomedical research. He was a Wellcome Trust Overseas Senior Fellow in Biomedical Science in India, and he is a Fellow of National Academy of Sciences, the Indian National Academy of Sciences and the Indian National Science Academy. He is also the member of prestigious organizations like International Complement Society, the American Association of Immunologists, the Molecular Immunology Forum, and the American Society for Microbiology. Professor Sahu expertise extends beyond research as he has been involved in patenting innovative solutions. Notably, he holds patents for peptides that inhibit the complement activation, which is approved by US FDA. He also holds a patent for a chimeric protein used in treating the pathological conditions involved in the complement system. Today, we delve into Professor Sahu's incredible journey, discuss the latest development in biotechnology, and gain insights into the exciting work happening at the RCB Faridabad under his guidance. So without a further ado, I extend my sincere and heartfelt welcome to you, sir. Namaste and thank you for blessing Beyond Shodh and providing us the opportunity to showcase your legacy to my audience.
Okay, namaste and thank you so much, Sonia, for having me on your show. I'll be happy to answer all the questions that you may have related to my research and my journey. Thank you so much once again.
You're most welcome, sir, and thank you so much for joining us. So, sir, to start with the episode, can you please share with us the overview of your research and vision of your laboratory?
Okay, so the research which I do is primarily basically the interactions of the viruses and the complement. Complement system is one of the innate immune system that is there in our body to fight various pathogens, and viruses is just one of them. There wasn't much known in this area, and that's the reason particularly what was not very clear when I started the research is how viruses evade this complement system and how critically this is really needed. So that's when I started looking into that, and then we figured it out that the successful viruses have various mechanisms to evade the complement system, and we have identified certain molecules which are there in different families. For example, pox viruses and herpes viruses. We have characterized them, we have understood how they function, and we have also tried to understand what happens if you disable these proteins from the viruses and can really complement attack. And what are the role of, again, from the complement side, in direct neutralization of the viruses as well as boosting the acquired immunity, which also it does it very well. So that's the overall kind of science which my laboratory focused on.
So, sir, what exactly happens or what all mechanisms are triggered when a particular virus attack a human cell? So, are there some canonical or some special mechanisms that are there?
Okay, so whenever our body is exposed to viruses, there are two main sort of arms of the immune system which come into play. One is the innate immune system and another is the acquired immune system. The job of the innate immune system is to take care of the viruses immediately, right? If the viruses come, they are there, they are ready to attack. So they are, in layman's languages, they are more like sort of a policeman, right? So they are there, they just fight it out. And the next job is that also they take this information, they gather the information and give it back to the acquired immunity. So if the innate immune system fails, then they pass on the information to a much sophisticated system, which is acquired immunity, right? And then they trigger this and they make specific immune response against these viruses. Acquired immunity can attack. And now because of COVID, we understand very well that the two main important components which are there in the acquired immunity is one is antibody, which we know very well, right? And the monoclonal antibodies was the one which was allowed and given, and also the convalescent plasma which was transferred, and also the T cells, right? Particularly the CD8 T cells which can attack, and CD4 cells are the T cells which can help again to boost this response, right? So these are the two sort of main, and in the innate there are many different components. Complement system is just one of the components of the innate where we focus.
Can you please tell us about your background and what exactly led you to specialize in this field of viral immune interaction and the complement system?
So my background, my studies, what I did is after the 12th, I did microbiology in BSc. So that's my basic training. And then I went on to do the master's again in the microbiology. And during my master's, I got exposed to immunology, which we didn't understand or studied so much in the bachelor's degree. And that interested me a lot, and I had a lot of questions to really figure it out. And there were no good, teachers, or I couldn't do PhD there because I did all this in Nagpur. So I came to know a professor in Delhi, University of Delhi, who was doing, immunology, and therefore I thought maybe it's a good idea. So I came to Delhi to do my PhD in the immune system, and then particularly that's the time I got exposed to the complement system. Part of my PhD thesis was focusing on this. So then I decided to focus more and really understand and take it. And then I did postdoctoral training in complement biology with two of the pioneers of this particular field. One is Professor Michael Pangman in UT, Texas, and another is John Lambris at UPenn. And then I came back, and because of my background in microbiology, I decided to focus on viruses because there was a good amount of work which was going on in how the bacteria evades the complement system. There wasn't much known really about the viruses. So I thought of focusing on virus and complement interaction. And that's how I decided to get into this.
So sir, what was your first viral system?
So I started working first with the pox viruses, right? So as we all know, pox virus, particularly everyone knows about the smallpox, right? Which caused, so many deaths. And the virus, again, which was used as a vaccine, was also pox virus, which was a vaccinia. So variola was the one which was a causative agent, and vaccinia virus was again the system which was, vaccine, which was used as a vaccine. So this is where, when I started looking for molecules which evade, I found that both these viruses, the one which is a causative agent and the one that was used as a vaccine, contain this protein. They differ a little bit. And I thought this is a very good system where I can figure it out how the one which is the pathogen, how that protein works, and the one which is in a vaccine, how does that work, right? And that's where I started working.
Okay, great, sir. So, sir, during your journey, how do you see that your family values, upbringing, and support influence your scientific journey? And also, who were your role models and mentors who inspired you along the way?
So, my family background, both my parents did not get much chance to really study. When my father was close to 10th standard, his father passed away, and he got into business because there was a need to support the family. But he always felt that, I should study more. So he told me, you study as much as possible. And he allowed me to study and never told me anything that I need to support the family. So I wanted to study, and I was always very fascinated by science. So I used to read even books which are not from my curriculum and go to library. So I used to spend a lot of time going to not my just college library, but also other libraries. When I used to go to, I'm basically from a small place called Varda, where the Sevagram is. Sevagram is known because of Gandhiji, because the ashram is there. And close to that is Nagpur. So I used to go to Nagpur, then go to different libraries, study, open the books and try and understand. So there's a lot of interest in how things happen. And when you start reading more and more, you figure out that many things are not known. Questions are there, right? And I didn't have anyone to ask questions. So I thought, if I keep studying, maybe someday I will find, the answers to that. So that inquisitiveness kind of, and my teachers were very good, right from my school days to my college, very, very supportive. And they helped me in everything, right from not just explaining things which I didn't understand, but also improving my writing. I was not good coming, from Marathi medium. I was not good in writing. So when I used to write, a lot of times it wouldn't make much of a sense in English. So they would help me in really writing. So they were really very, very good. My mentor was very good. And when I went for PhD, again, I was always blessed with very good teachers. And he did his PhD in Philadelphia and UPenn and came back. He did PhD with, he had great students. Kunal Saha. So Kunal Saha was my mentor. He did his PhD in the US, came back, to India and started serving. He was in Panth, and then he moved to DU. And he used to teach me on a daily basis. He would give me some book and say, read it. And the first thing he would come is, okay, tell me what did you read? And he would ask questions, and that kept me on the toes that, I have to study. And he trained me a lot. And even my mentor, first mentor, Mike Pangban, was almost like him. He would make sure that I understand, discuss on a weekly basis. He used to come specifically on Sunday to discuss with me. Because, although he was Christian, and his family used to go to church, and he thought it's better that he discusses with me on Sunday. So it's to make sure that he comes on Sunday just to discuss. So yeah, and we used to have like hours long discussion, maybe till lunch or so, and then he would move. So yeah, I was always blessed with great mentors, and these were the role models to me, and they were the one sole reason why I'm here. Otherwise I wouldn't have been here.
So sir, in your lab also you follow their research practices or?
Yeah, so I follow whatever I learn. So I learn many things which meticulously one should document. So I try and teach this to my student and take them. And in India, our students are not really exposed to research. Right? I got never exposed to research until I joined PhD. The same thing is true. So I always made sure that my student discuss with me on a daily basis. So I used to discuss with them one-to-one, make sure that they understand what they are doing, make sure that they read the paper and try, understand that, and also come up with the experiment. Slowly I would tune them to come up rather than me telling them is do it like this. I always believed that if they start thinking and coming back, and I never ever told them to do anything which they didn't understand. So I made sure that they do, and I think if you talk to them, hopefully they will tell you the same thing. Sometimes I used to be tough with them when they were not
Sometimes
reading.
It's necessary.
Yeah, and but then my whole intention was that they really understand what they are doing. And I think in PhD, it's not about what exactly you are doing. It's more about understanding things, what you are doing, right? And really understanding how to do research. It's more like a training to do research rather than making a big discovery. That's what I believe in, is that, and then I tried my best to the best of my ability. I gave them, the training which I thought is good for them.
Yes, great, sir. So, sir, as we all experience that COVID-19 pandemic has shown us how viruses, the simplest and tiniest form of life, can pose a serious threat to humans, which is the most evolved and complex species on Earth. So, how do you view the role of technology and its advancement in this domain? And are we well prepared to combat the future viral invasions, or should we be worried about that some viruses may
No,
came
so
and
the couple of things which we need to understand and which we understood again very well is, one is that the viruses that can really cause problem to us, they have to be novel viruses, right? Unless that we are never exposed, most population is not exposed. So although new viruses, we keep getting new viruses, when we get exposed, and they many a times come, they are zoonotic viruses which they come, there are many things they require, and therefore not only that they should replicate well, but they should survive in the environment. They should able to cause the problems, disease in humans particularly, right? So there are a lot of requirements, and therefore we don't see these kinds of pandemic every year, although we know that viruses are mutating as and when we talk. The next question, biggest question was whether we are well prepared or not. So we do have vaccines, but we can never ever have vaccine for something which is unknown, right? So that's always a problem. So even if the disease is in one particular pocket, usually we would have vaccine for that, but the disease is not there, or the virus is completely novel, as that occurred in case of SARS. We didn't have anything, but then what we really had, the science did, is we had many platforms, which where they were there, right? So adenoviruses were there, we had the mRNA platform which was there. All these allowed us to make the vaccine very fast, so it was done in record time, right? So we do have platforms, and there are other platforms also which people are trying and develop, and these are now well tested now because of COVID. So in my opinion, even if there is a virus or new pandemic that happens, we'll have these platforms, and by that time we'll have many more platforms which people are working on, which will be there. And therefore I think vaccine which you make, and which used to take years and years together, that time has been cut short, right? And a lot of the regulatory process have also been modified because we have to get them done in a record time. So all these we have learned, so we have learned these lessons, and I think these will really help us in facing the new pandemics as and when it comes.
So, sir, how long is exactly the process of identifying this virus and in the development of vaccine?
Yeah, so see, what people look for is something, a protein. Let's say if we are talking about, originally, people used to use just the entire virus, but now we look for what we can target, and such target molecules can be one or more than one, right? And these are not very difficult to identify. When can look at the response that is getting, and if you target that, whether that would neutralize the virus or not. And the entire process, which could be years, is now dropped down into months together, right? So in few months we will be able to do. Of course, that requires the entire setup to do that. So in a normal laboratory it cannot be done, but if you have a setup to work, particularly in the industrial setup, that would be something which can be done very fast.
Great, sir. So, sir, are there any particular challenge or obstacle that you have encountered during your journey that you would like to share, or how you have overcome that?
Well, honestly, I never really faced any major obstacle. The only obstacle which I faced is during transitions, or sort of, when I was doing my master's. And that time I was very keen in doing immunology. In those days, it was not the era where you could Google it,
Yes,
or
yes.
go and just figure it out how many labs are there and where I can go, and simply from your mobile, send a text message to them and saying, I would like to do, or all the information. This is information age which was not there. So during my time when I was looking for, there was very, very difficult. There wasn't even literature available. How many immunology labs are there? And therefore, what happened that I used to just, talk out to people saying, this is what I am interested in, which I do it even now. You never know who can help you.
Yes.
So one of the persons from Delhi came to Nagpur, and people knew that he is looking to do some PhD in immunology, and that's how they said, oh, you go and talk to this person. So that was the time I didn't know. I know it was kind of a challenge, and I wasn't sure whether I'll be ever able to find out a lab where I can do PhD. So that was my biggest challenge. After that, when I did PhD, by that time, we had enough resources in Delhi to figure it out, where to go for postdoc and how to find those, and how do you find labs. So there, things were much, much better, obviously, in Delhi, and compared to from where I came from. So yeah, that was the sort of, I would say, hurdle. But as I said, I was blessed with great teachers, and therefore it was very smooth sailing all the way along.
Recently, one report came on the number of retractions and errors in the publication from India, and that bar is going very increased as compared to previous years. So what exactly is going wrong with the research community? Why so much of retraction, so much of error? Somewhere down the line we are not getting in a correct way. Maybe it is the load of getting more quantity of publications rather than focusing on the quality.
Yeah, see, there is one thing which students need to understand, and others who are in research know, or if they don't know, then we need to understand that it's not a question of how many number of papers you publish, right? It's not the volume that matters. What matters is, are you finding a new information? Are you adding to the knowledge, right? And it's simply the joy of finding something new is what drives innovations and the research, right? So that joy is very important. It's not about how many papers you're publishing. And if you try and just think about paper, and if you put unnecessary pressure, right, on people, that may lead to sometime these kind of, people, sometime we see that they are fabricating data. Maybe they are told that you have to have enough number of papers and this, but this is not really the, right thinking. And there is somewhere this wrong information is maybe floating, which in academia no one ever says. If you go to talk to any teacher, any professor, they will never ever tell you that you need to have certain number of papers, right? Or even the papers in certain journals. That is not which is critical. What is critical is that have you learned how to do research? That's when you are doing PhD, it's all about learning how to do research, right? That's what is more important. The journey is more important than really publishing 10 papers, right? And that's what one should focus more on. Do I really understand? Do I understand how to ask questions? What are the ways of answering those? Is there only one way, or can I answer that in a multiple ways, the same question? These are the things which you want to learn. So, I would like to give through your podcast this message to everyone. It was never, and even now it is not about the number of papers or even the journal. Nobody tells you that if you have a PhD, you should publish in a Science or Nature or this. If you do it, fantastic, wonderful. That means that you have discovered something very, very novel, and therefore you are there. But simply mere the process is important, and it doesn't matter where you publish, and therefore never ever fabricate the data, right? So I want to tell all the students very clearly, do not get into this. Don't try and fabricate. It can ruin your career, right? And that's not why you are here. You are actually here to enjoy the journey and learn to do the science, right? And not to really have 10 publications to do this. So that's, I think, is completely wrong way of thinking.
Because sir, you are correctly elaborating this point, that because research comes with the responsibility, and it's the responsibility of the researcher that to put out correct publication, put out correct results, so that based on his research, the other upcoming research will again,
Yes.
that is a saying, the garbage in is garbage out.
Correct.
So it's again, this is the responsibility of a researcher to put out correct information, and not just with the sake of publishing more number of papers. We should not support false information. So coming to your research, sir, what potential breakthroughs and discoveries do you find most exciting and promising for the future application in the field of virology and immunology?
Yeah, so, there are many, of course, but then the one which stands out, which really helped the field, and we have seen is in COVID, is the platform for the mRNA, right?
Okay.
This was a game changer. Nobody thought, and it has cut down. And the advantage, the biggest advantage is that irrespective of the pathogen we are talking about, we can, of course, it has many other also advantages just from apart from vaccine, but this technology really stood out, right? In this that and of course we have seen that the Nobel Prize is given for that.
Yes,
So to me, if you ask one that made a significant dent in the recent time, I would say is that this mRNA technology in the vaccines, which can record time and saved so many lives that the world would remember this particular change that occurred during COVID.
So, sir, what are the key questions or areas of research that you and your team are most eager to explore in coming years?
Yeah. So we started with identifying molecules that help the viruses evade, right? And then what we started now looking more into that, if these are not there, then what happens?
Okay.
How the complement controls this? What are the crosstalks of the complement system with various different pathways, right? So all the pathways by and large, they evolve, they co-evolve, right? So it's not a good idea to think that, they evolved in isolation. Therefore, they must crosstalk with multiple things. How do they crosstalk, right? And if we really understand this crosstalk, I think we will understand not just how complement directly works or acts, just with the T cells or with this or with that, but in totality, when we understand the intricate pathways that are linked, and that is something I think would be very, very important. And that's where we want to focus, that not just the role of complement directly acting on the virus, or not just complement talking with one or two pathways of the acquired immunity, but the overall impact and the entire crosstalk. That is where we are trying to focus more on.
Great, sir. Now, as a director of Regional Center for Biotechnology, what is your vision and policies that you want to strengthen for advancement of the innovation and technology within RCB, and to make it one of the leading center of India in the area of biotechnology?
Yes, so there are many components which any institution requires if we have to excel. The most important foremost thing is, of course, the faculties which you need. You need a critical mass. Not only you need a critical mass in overall in an institution, but also critical mass in the area you want to really progress in, right? So that would be the primary goal, is to have critical mass. We don't want to focus obviously in too many places, but wherever we want to focus, we should have critical mass in that. And therefore, one of the things is, of course, have enough number of faculties and recruit more people. So and this is an ongoing process. You keep need to recruit because science is progressing.
Yes.
And therefore, it's not about hiring everyone at one time, but keep hiring and keep doing, get new technologies, get new blood and new thinking all the time. That's one component which where we want to do, right? And virology is our core strength, so definitely focus on that, right? That would be one of the things. Cell biology is another thing where we are doing, and actually the interaction of the viruses in the cell, that's something, which is there. Also in the plant, we are good in plant biology, and also we are focusing more into how the plant pathogens, particularly viral pathogens. So from that side also we would like to do. So these are the areas, and of course for doing all that, one needs different platforms, the high-end equipments, right? We do have some. RCB has good ATPC facility, but also strengthen this, right? And also strengthen the manpower in that area where you can get the best of the technical people, staff to help you to do the research in record time. So that would be another one. Also, expand. We have bioincubator here, right? To even expand this so that people who would like to incubate, they can incubate, make this facility much more, more robust, have more things for the students. And RCB's mandate, as you know, is not just research, but also education, right, and training. So we would like to have many more training programs started, and also, think about trainings which people need even after their degree to get into particular jobs. So can we create certain courses for them, certain trainings for them, which can help them to get job where they like to do, and also education, right? So we have, masters, MS, PhD student here. So we provide MS degree, we provide MS, PhD, we have separately PhD. But, there is also something which RCB is going to get into, that is have a larger PhD structure, right? PhD program, where we are thinking along with the DBT, all the DBT institutions coming together. So by and large, most DBT institution barring RCB and ICGB will come now under the umbrella called BRIC, right? Yes. And so we want to have a BRIC RCB PhD program where PhD students, right, which we are thinking to start with something, a good number, close to 100, okay, and then expand further, right? So have where the faculties, the best of the faculties from all the institution can come teach, provide the training, right? There are a lot of things which are under discussion. Now nothing is final yet, and therefore I don't want to go through the details of those because these are not finalized. But I would like to certainly add that there will be a wonderful PhD program, one of its kind, which will, which will be done in RCB to start with, where the students will get their trainings here for 6 months, both, not just the theory aspect of the subjects, not just the coursework, but also the hands-on, the best of the hands-on, and then they will go to their respective institution with the DBT institution. So there will be some kind of this, large PhD program where, all the DBT institutions are going to come together to do this, and that will be initiated in RCB, and the training will happen initially in RCB, right? So that's something, the program, and we are still thinking more and more, and that depends on, how many programs we can do. I have many things in mind, but unless, they become a reality, I don't think it's right for me now to just discuss those, because we are trying certainly many things to initiate, but let's see, and you will know as and when they come.
Yes, yes. Great, sir. This sounds very interesting idea, and large number of students will be involved to get very good training on hands-on training, specifically hands-on training. So, sir, as we all know that we in previous years and now we are making tremendous progress in research infrastructure and funding in recent years. Uh, Recently, the NRF also declared. So, what are the gaps that Indian science needs to fill in order to compete with the global leaders in the scientific research? Are the funding is appropriate, or still we need more amount of GDP to be pushing into the research and innovation?
Yeah, see, this is a policy matters, right? A lot of structural changes need to be done. If you see globally, it's not just the government that puts the funding, but majority of the funding comes from the private sector, right, in research. That is something which government is trying now, that through NRF that this will be done. And I think unless this happens and more amount of money comes into research, the mass which is there, critical mass, is not enough, right? We need many more people and many more institutions to really make an impact significantly if we have to do that. So even in an institutions, the number of scientists are not that many. If you compare that to anywhere overseas, when you talk about a research institution, you talk numbers in hundreds, right? And those kind of, and of course for so many faculties and the researchers and the scientists to be there, a lot of funding is required. So for that, there has to be a significant component, much more than the government component, which should come from the private sector. And NRF is meant to do that. So let's,
finger crossed.
Keep our fingers crossed and hope that, we'll be able to do that. But I think it's a significant step forward, which is done, and I think it will lead to fruitful results. I'm very positive about it.
And sir, as you are talking about critical mass, and most of our the young generation, they are in the university system, but it is not happening that the university infrastructure is research friendly, or the kind of ecosystem that is needed for research is not supported. So how can these big institutions or these big institutes can support these smaller, in maybe in some programs where they allow the researchers for 2-3 months carry out their research and then go back?
So there are a lot of schemes available, right? So it is not that there are no government funding or other fundings available for the universities. Now there are a lot of fundings which are available. These, of course, are competitive in nature, and there are a lot of mechanisms as well. The government has made, or the funding bodies have made, right, which are available for them. And it is also very easy nowadays for any researcher actually to go from one place to another, right? If they want to, let's say tomorrow someone wants to come to RCB, a faculty who is there in some university, and they want to align with something, we encourage that. And of course, almost all institutions would encourage that. So I think it's more up to the faculties to take this opportunity and try that. And use the fundings which are available, right? These are there. Almost all funding bodies give money, right, to this. So utilize this opportunity, I think. So I think it's more maybe, awareness to the university
Outreach.
that what is available out there, and also a push from their side as well, right, which is required. And I think if that happens, it will definitely, it's not that nothing exists. A lot of things exist which are already there. I think maybe the information is not getting trickled down to that level for whatever reason. So I think it's people like you who can, give this information is what are all the schemes which are available to them, right? And in such kind of forums, if you tell all the schemes, I think that would help them probably to go and apply for those and get those, right? So there are a lot of schemes,
So what is your advice to the budding scientists who wish to carry forward their research journey?
Yeah, so be passionate about what you do, right? And if you are passionate about what you do and you really love doing, you will excel. There's no one who can stop, right? But early on, find out whether research is really what you really like to do, right? Whatever you are doing, very, very important to find out for you, because otherwise, you will end up finding that, oh, why I'm spending so much time?
Yes.
There is so much life out there. My friends are doing so many things. I'm sitting in this lab and watching at a
They
test
are
tube,
earning more
right?
than me.
They are earning more than me. So in a research, it's not about money. It's a passion, right? It's a passion to find new knowledge. It's passion. So when you find something new, you get so excited, and same thing for the PIs actually, when the students find new things, and you can imagine your student going on the podium up there in an international conference and giving a talk, and no one in that audience knows about that. Your lab and your student is the one. So think about the high they can get, that I'm the one who is telling you this is the way it works,
Yes.
right? That high, I think, is what keeps all the students and all the researchers going. And it's not about money, right? And money, I think, to me, at this age, I think money is just there for sustenance, right? And it doesn't give you kick. It never gave me kick. Maybe some people it does give kick and they want to make a lot of money. But I think to me, it never mattered. As long as I get enough money to, get going and survive, I think that's more than enough. So if that's the way you think, that you really love and that's what you really like to do, that's your cup of tea, and go after it. And if you are passionate, you're going to excel. Nobody will ever stop. And I have seen people coming from all over the places, from places where they had nothing, right? And they did so well, and they reached places, right? And you see, you see and you read it in the newspaper.
Yes.
The Indians are doing great everywhere, right? They are on the top. Why they are there, right? They could compete. They just love doing what they want to do. So I think it's all about whether you are passionate about things. That's my thinking, right? So that's what I would say.
So, sir, are you satisfied with your journey? So, what is one piece of wisdom from your journey that you want to
Yeah,
share?
no, so I think I'm very, very satisfied. I loved what I did. I always thought that whatever I do, apart from publishing, if at some point, if it gets translated and helps, human life, would be something which would satisfy. And I discovered something during my postdoc, which of course a lot of people worked on it. It's not initially I found something very interesting.
Sir, if
A
you can
molecule.
just tell us a little bit about that.
Yeah, so this is something about we were looking for. um there's a molecule called complement C3, which is a central molecule. Now we were trying to see if we can regulate this molecule, right? The idea was not really to find any inhibitor against this. The inhibitor to really understand the basics of these molecules, how you can regulate this molecule. And I came up with some molecule which looked very, very good. And I had earlier worked on the same problem in my previous postdoc. I knew, and I didn't get anything significant there. So when I got this, I was just very happy to find this. And then, we in US, it's always people like to patent it. So it was filed, but then I kept working for another 4 or 5 years on that. Then came back to India. And a lot of people worked. It was picked up by companies. They started working on it, and finally it became drug, and it went into clinic, right? And that it is doing better than the earlier drug, which was there was only one drug in the whole world available, which was a monoclonal antibody in the complement system. This in one particular disease, did very, very well, which was tried into that particular disease, did excel, did really well. So I was very satisfied that something which I did.
That feeling is,
Yeah.
you know,
So it's not, like I said, it's not about money, right? So am I satisfied? Yes. And we did some good work here, which was very well appreciated, outside, and people recognize our lab for that. Of course, it's all about the student and their hard work. I was just guiding them to do, and one thing led to another, and students kept coming and kept working on that. So yeah, so when someone talks about the complement and virus interaction, they always, our lab is there as one of the leading labs which they recognize, and that's a great satisfaction that even after coming back to India, I could do something interesting. I could teach and train students. We could do some really good work, which everyone recognizes that it is a good work, right? So this is, I think it's a good satisfaction. It's great, which did, and I don't regret a bit that, and I'm very, very satisfied. I loved my journey. So when I look back, I'm very satisfied person. Of course, there's a lot to be done. So don't want to stop here. Keep working. But yeah, it was very satisfying.
Very well said, sir. And we know that you have whole lot of to-do list,
Yes.
which you will definitely accomplish. So thank you so much, sir, for your time and sharing your thoughts and your journey. Thank you so much.
Thank you so much for having me. Thank you.